Hi, today we are now going to see about some more subtopics in the biotechnology. So, vaccines. Vaccines play an important role under biotechnology. So, what are the vaccines? So, we use vaccines to prevent diseases before it comes. So, it is the idea of using the vaccine. So, vaccine what it contains? Okay, we knew about the polio vaccine, and there are so many vaccines are there. So, vaccine contains less virulent or killed pathogens. So, vaccine gives you a active acquired immunity to a particular disease. So, we have to prevent the disease before it comes. So, in our body, antibodies will be produced. So if the foreign body which means antigen comes in our body have to be ready. So the antibody which was produced in our body will fight against that. So we we don't get that this is the idea of creating a vaccine. So vaccine is nothing but which contains less virulent or killed pathogens inside it to prevent any diseases. So it will give you active acquired immunity to a particular disease. Okay, this is a small information you have to remember. After vaccines, you have to remember this kind of information. So the first rDNA, recombinant DNA, was constructed by Cohen and Boyer in 1972. Okay, this is very important information you have to remember. The first recombinant DNA was constructed by Cohen and Boyer in 1972. What they did was they isolated antibiotic resistance gene from Salmonella typhimonium and they injected the antibiotic resistance gene into the E. coli. You knew about vector. Vector means um, it will carry the substances which have to be injected to the the recipient. So E. coli here is a recipient. So we are taking the antibiotic resistant gene and vector is like it will carry it and give it to the E. coli. So when the plasmid of the E. coli attaches to the E. coli, rDNA will be synthesized. Recombinant DNA. Recombinant DNA, which means its genetic makeup is different from the normal. Okay. So and then I will tell about enzymes. So first we are going to see about lysing enzymes. What is my lysing? Lysing, which means some breakdown or digesting. So we have to break down it and digest it. So lysing enzymes, we are under enzymes topic. First we have to see about lysing enzymes. So example, in plant cell we knew that cell wall will be present and then plasma membrane will be present. Then only we can reach the DNA. Okay, so the cell wall of the plant cell which contains cellulose. The idea is we have to reach the DNA. So if we have to reach the DNA, first we have to enter inside the cell wall, then we have to enter into the plasma membrane, then only we can reach the DNA. The cell wall of the plant cell which contains cellulose, we have to digest it. So the enzyme we have to use is cellulase. Using cellulase enzyme, we can break down this cellulose. So we break down, we enter inside the cell wall. Then what we have to do? Then plasma membrane. This is plasma membrane. You know that a plasma membrane contains phospholipids and proteins, plasma proteins. So what we have to do is, okay, plasma proteins, sorry, phospholipids and plasma proteins. So we have to digest this phospholipids and plasma proteins. So how we can break down this? So proteins, so we have to use proteases to break down this, and phospholipids we should use lipase, phospholipase or lipase. So we should use phospholipase. So these two enzymes we have to use. This will comes under lysing enzymes because it is breakdown and digesting the particles. So then only we can enter into the DNA. So this type is lysing enzymes. So and then under lysing enzymes, animal cell. Animal cell, it don't have cell wall, only plasma membrane. So we should use the phospholipase plus proteases. Okay, so we should use the two enzymes we have to use. And then fungal cell. Fungal cell, the cell wall contains is made up of chitin. Cell wall is made up of chitin, and then plasma membrane, and then DNA. So the cell wall and then plasma membrane and then DNA. So the cell wall is made up of chitin, so we should use chitinase to digest chitin. And then plasma membrane, so we have to use phospholipase and proteases because plasma membrane is made up of phospholipids plus plasma proteins. To digest proteins we have to use proteases, and phospholipids we have to use phospholipase. That's it. And bacterial cell. Only animal cell is the exceptional case which don't have cell wall, but all other organisms are having the cell wall. Bacterial cell wall is made up of peptidoglycan. So peptidoglycan. So cell wall which is made up of peptidoglycan. So we will use lysozyme to isolate the DNA. That's it. To digest it we have to use lysozyme. So these are the lysing enzymes. Okay. Plant cell, it have cell wall, plasma membrane. So cellulose, the cell wall is made up of a cellulose in the plant cell. So we will use cellulase. The plasma membrane, I already told about that. And the cell wall is made up of a fungal cell. Cell wall is made up of chitin, so we can use chitinase. And bacterial cell, so cell wall is made up of Cell wall is made up of peptidoglycan, so we should use lysozyme. So that's it. Why animal cell? Animal cell, we can't eat the plant, um, because we can't eat the plant because we don't have the cellulase enzyme to digest that. This is small information I told you about that. Okay, and then the second type of enzymes I will tell in the second video.